Let's explore the calculation of duplex distance between two short DNA sequences. The duplex distance essentially quantifies the dissimilarity between two DNA sequences, focusing on the number of non-complementary nucleotides when the sequences are aligned. To calculate the duplex distance, there are typically two primary steps. Firstly, determine the number of complementary base pairs between the two sequences. Secondly, calculate the duplex distance using the formula duplex distance equals the number of non-complementary nucleotides. Now let's consider a specific example. Suppose we have sequence 1 which is 5' Ag3' and sequence 2 which is 5' TCGA3'. Our goal is to calculate the duplex distance between these two sequences. Step 1 involves determining the number of complementary base pairs. In this case, adenine or in sequence 1 pairs with thymine or T in sequence 2, and guanine or G in sequence 1 pairs with cytosine or C in sequence 2. Therefore, there are two complementary base pairs, a pairing with T and G pairing with C. Step 2 involves calculating the duplex distance. The duplex distance is defined as the number of non-complementary nucleotides. In this specific instance, there are 0 non-complementary nucleotides because all the nucleotides in the two sequences are complementary to each other. Consequently, the duplex distance between the two sequences is 0. Let's examine another example to further clarify the concept. Imagine we have sequence 1 which is 5' Ag3' and sequence 2 which is 5' CAG3'. Again, we want to calculate the duplex distance between these sequences. The first step remains the same, determining the number of complementary base pairs. Here, in sequence 1 pairs with T, even though the T is not explicitly present in sequence 2 are to corresponding position. G in sequence 1 pairs with C in sequence 2. Thus, we still identify two complementary base pairs based on potential alignment with T and G with C. Now, let's move to step 2, calculating the duplex distance. Using the same formula, the duplex distance equals the number of non-complementary nucleotides. In this scenario, there is one non-complementary nucleotide, which is the T in sequence 1. This T does not have a corresponding complementary base in sequence 2 when considering a reasonable alignment. Therefore, the duplex distance between these sequences is 1. The duplex distance essentially highlights mismatches or unpaired nucleotides when you try to align two DNA sequences. A lower duplex distance implies a higher degree of complementarity, while a higher duplex distance suggests greater dissimilarity. Now let's dive deeper into the implications and applications of duplex distance calculations. In the realm of molecular biology and genetics, understanding the complementarity between DNA sequences is fundamental to many processes. For instance, DNA hybridization, which is the process of joining two complementary strands of DNA to form a double-stranded molecule, relies heavily on the principles of base pairing. Similarly, polymerase chain reaction or PCR, a widely used technique for amplifying specific DNA sequences, depends on the accurate binding of primers, short DNA sequences, to the target DNA. The duplex distance calculation can be a valuable tool in these contexts, allowing researchers to predict the stability and efficiency of DNA hybridization and primer binding. By quantifying the degree of complementarity between two sequences, we can estimate the likelihood of successful hybridization or primer annealing. A lower duplex distance, indicating high complementarity, suggests a greater probability of strong and stable binding. Conversely, a higher duplex distance implies a weaker interaction and a lower chance of successful hybridization or annealing. In addition to these applications, duplex distance calculations can also be employed in the design of DNA probes for diagnostic purposes. DNA probes are short, labeled DNA sequences that are used to detect the presence of complementary sequences in a sample. By carefully selecting probe sequences with low duplex distances to the target sequence, researchers can enhance the sensitivity and specificity of their assays. Furthermore, duplex distance calculations can be applied in the field of evolutionary biology to study the genetic relationships between different species. By comparing the DNA sequences of various organisms and calculating the duplex distances between them, scientists can gain insights into their evolutionary history and relatedness. Smaller duplex distances generally indicate a closer evolutionary relationship, while larger distances suggest a more distant relationship. Now let's consider some additional factors that can influence the duplex distance calculation. One important aspect is the length of the DNA sequences being compared. Longer sequences are more likely to have non-complementary nucleotides simply due to the increased number of opportunities for mismatches. Therefore, when comparing duplex distances between different pairs of sequences, it is important to take into account the length of the sequences. Another factor that can affect the duplex distance is the presence of modified bases in the DNA sequences. Modified bases, such as methylated cytosine, can alter the base pairing properties of DNA and influence the duplex distance calculation. In some cases, modified bases may increase the duplex distance by preventing proper base pairing. In other cases, they may decrease the duplex distance by stabilizing the DNA duplex. Furthermore, the environmental conditions, such as temperature and salt concentration, can also affect the duplex distance. Higher temperatures and lower salt concentrations tend to destabilize DNA duplexes, leading to an increase in the duplex distance. Conversely, lower temperatures and higher salt concentrations stabilize DNA duplexes, resulting in a decrease in the duplex distance. It is important to consider these factors when interpreting duplex distance calculations and comparing results obtained under different conditions. To summarize, the duplex distance calculation is a useful tool for quantifying the complementarity between DNA sequences. It has a wide range of applications in molecular biology, genetics, diagnostics, and evolutionary biology. By understanding the principles and factors that influence the duplex distance, researchers can effectively utilize this metric to gain insights into DNA interactions and sequence relationships. The duplex distance provides a simple numerical representation of the differences between two DNA strands, allowing for comparison and analysis. The calculation focuses on the number of mismatched base pairs, providing a straightforward measure of dissimilarity. In practical applications, the duplex distance is often used in conjunction with other sequence analysis techniques to provide a more comprehensive understanding of the relationships between DNA sequences. For example, it can be combined with sequence alignment algorithms to identify regions of high similarity and regions of high dissimilarity. It can also be used in conjunction with phylogenetic analysis methods to infer evolutionary relationships between organisms. In conclusion, understanding and calculating duplex distance is a foundational skill in molecular biology and related fields. It provides a simple yet powerful way to quantify the degree of complementarity between DNA sequences, enabling researchers to make informed decisions about DNA hybridization, primer design, probe development, and evolutionary analysis. The concept is simple to grasp, yet its implications are far-reaching, impacting various areas of biological research and applications. When working with DNA sequences, always consider the duplex distance as a potential metric for understanding their relationships. Its simplicity and versatility make it a valuable addition to the toolbox of any molecular biologist or geneticist. The calculations provide a quantitative measure that can be used for comparative analysis and hypothesis testing. Always ensure that you clearly understand the context and limitations of the calculation when interpreting the results. Remember to consider factors such as sequence length, base modifications, and environmental conditions, as these can influence the duplex distance. With careful consideration and application, the duplex distance can be a powerful tool for unraveling the complexities of DNA sequences and their interactions.